good morning. My name is Akshay Sharma, and today we'll discuss about the process of photosynthesis. And specifically in photosynthesis, we'll discuss about the mechanism of light and the dark reaction. Now, as you can see in the definition of photosynthesis is defined as the process of converting light energy to chemical energy and storing it. Storing the energy in the bonds of sugar. As you may have learned in your lower classes, photosynthetic equation is somewhat like this: six molecules of carbon dioxide combined with six molecules of water. Right, in the presence of this reaction occurs in the presence of sunlight or any source of light, along with the help of a molecule known as chlorophyll. Chlorophyll, you must remember that it is the pigment which is responsible for the green color of leaves and the stems in some cases. So, in the presence of chlorophyll and a source of light, these two reactants together form a molecule of glucose. This is C6H12O6. This is a molecule of glucose, and in the process, six molecules of oxygen are liberated out. This is a simplified reaction. Actually, the reaction also gives out some unused molecules of water. Uh, and there's one more thing that you must remember, being the students of biology, that in some books it's written that energy is given out during photosynthesis. You must remember photosynthesis is an endothermic process. It requires energy for its completion. It does not evolve any type of energy. The glucose which is formed in photosynthesis can be used as a source of light, can be used as a source of chemical energy in the process of respiration to form ATP molecules. But photosynthesis itself is an endothermic process. So let's get to the next slide. Uh, like you all know that photosynthesis requires, or any chemical reaction being, photosynthesis is a chemical reaction, and any chemical reaction, like all others, requires some conditions, some reactants, and some enzymes to, or catalysts in a case of chemical reaction, to take place. Right, so what are the conditions which are specifically required for our process known as photosynthesis? See, photosynthesis requires, the first thing, a molecule of chlorophyll. Now we all know that every green plant has chlorophyll molecules. Chlorophyll A is present, chlorophyll B is present, then C, D, E, and F. All these types of chlorophyll molecules can be present in a leaf. Apart from that, you must have learned that bacteria, some of the bacteria are also photosynthetic, and those bacteria contain bacteriochlorophyll. Alright, so chlorophyll is the essential requirement for photosynthesis to take place, because this is the molecule which actually absorb the light energy, whether it is coming from sun or any other light source, and this is the energy which is further converted into chemical energy with the help of all the other reactants and all the other conditions. Now, having the amount of chlorophyll or the type of chlorophyll is an intrinsic property of a leaf or a plant, and it does not change much, and thus it does not affect the rate of photosynthesis very much. So we'll move on from chlorophyll to the next important condition for photosynthesis, which is the light source. Okay, actually, for any type of photosynthesis to occur, light is very important. Without light, photosynthesis cannot occur. Although we'll see later that some part of photosynthesis can take place and does take place during the night time when there is no light. But photosynthesis as a whole, the process cannot take place without light energy. Now, this source of light can be your sunlight, or the source can be the sun, which is true for most cases. But sometimes, as you see, some plants even grow in laboratory conditions, or some plants grow indoors where there is no direct sunlight falling on them. So, in those cases, the artificial sources of light, like your tube lights, your bulbs, your lamps, all these sources emit visible light, and they can help in photosynthesis. Now, regarding light, there is one more important thing, that there are two parameters of light upon which the rate of photosynthesis depends. The first is the quantity of light, the intensity of light. As you increase the intensity of light, the rate of photosynthesis also increases. right? If you get more food, you can get more energy. So light is a type of food. Photons, which the light has, or the packets of light energy, are required by the plant to perform photosynthesis. So more the photons, better the rate of photosynthesis, right? And vice versa is also true. So this is the quantity or the intensity of light. As we go on increasing the intensity of light, the rate of photosynthesis also increases. Now the second most important parameter regarding light is the quality of light. By quality of light, we mean the wavelength of light, and uh, many students expect that green light would be the best light for photosynthesis. Actually, it is the opposite. Green light is the worst light or the worst color for photosynthesis to take place in green colored plants. That is because chlorophyll themselves are green in color. That means they reflect all the green color that is falling on them. So the best color for photosynthesis, or best light for photosynthesis, is red light, followed by blue colored light, right? And the combination of red colored light and blue colored light is the best combination for photosynthesis, at which the rate of photosynthesis is maximum. Now then we move on to water. Water, as we've seen in the reaction preceding this page, the reaction of photosynthesis, it is one of the reactants, and without water, photosynthesis cannot take place. If water is very low, the rate of transpiration decreases. That further, in order to decrease the rate of transpiration, the plants close their stomata, and it further stops the exchange of gases. So in the absence of any gases, in the absence of any carbon dioxide entering the leaves, photosynthesis cannot occur. And if the water is too high in the the soil becomes waterlogged, and the cells of the roots cannot respire, and thus they negatively affect the rate of photosynthesis. Then the next point is optimum temperature. An optimum temperature range of say 25 to 40 degrees Celsius is required for photosynthesis. The rate of photosynthesis itself increases when we go from 25 to till 40 or 45 degrees Celsius. Above that it decreases, and below 25 degrees Celsius the rate of photosynthesis also decreases. Although this is not an objective point or not an objective condition for photosynthesis, because the optimum temperature can vary from a plant to plant. For example, conifers growing in cold climates will have a lower optimum temperature for perfect photosynthesis, while a plant growing in a desert condition will have a higher range of optimum temperature. So I think you get the point, how optimum temperature varies from plant to plant. Now the final and one of the most important conditions for photosynthesis is this carbon dioxide. I'm sorry. Right. So carbon dioxide is one of the main reactants. It is the source of carbon in glucose, although not the source of oxygen, which is liberated out during photosynthesis. It is the source of carbon present in the glucose molecules. So as we increase the carbon dioxide, or the amount of carbon dioxide present near the plant for it to perform photosynthesis, the rate of photosynthesis also increases, because the more the reactants, more would be the rate of photosynthesis, right? You must have learned in chemistry that this is the way chemical reactions behave. The more the reactants, the better the rate of photosynthesis, right? And if the quantity or the concentration of carbon dioxide in the immediate vicinity of the plant is very low, the rate of photosynthesis will also follow, and it will decrease accordingly. All right, so next we'll move on to the next slide, the different phases of photosynthesis. In class 9 we discuss about the chloroplast, or the kitchen of the cell. We call chloroplast as the kitchen of the cell. Now this is the time to learn why they are called the kitchen of the cell. Phases of photosynthesis are basically two phases. The first is the light dependent reaction, as you can see here, and the second phase is the dark reaction, which is independent of light. The light dependent reaction is also known as the light reaction itself. It is known as the light reaction because it cannot occur without the presence of light. That is why it is known as light dependent reaction, right? So it occurs in grana of chloroplast. Grana, if you remember, are the stacks of coins, as is mentioned in different books, stacks of coins present in the chloroplast, which are joined together by different structures known as stromal lamellae or stroma thylakoids. And within these grana, in the membranes of these grana, are present chlorophyll molecules. So this question regularly appears in many competitive exams. Where does chlorophyll molecule, where is chlorophyll molecule located in a chloroplast? So the answer it is located specifically in the inner membranes of thylakoids, which stack together to form these structures known as grana. Right. So light dependent reaction naturally occurs in grana because they are the sites where chlorophyll is present. And there are two types of light reaction. One is non-cyclic photophosphorylation and the other is cyclic photophosphorylation. Now what does this weird term photophosphorylation means? Photo means light and phosphorylation means addition of phosphate groups. So if the phosphate group is added in the presence of light in a non-cyclic manner, the process of light reaction is the non-cyclic photophosphorylation, and if it does occur in a cyclic manner, it is the cyclic photophosphorylation. Okay, this we'll discuss in the next slides. Then the next phase of photosynthesis is the dark reaction. Now dark reaction is not called dark reaction because it occurs in dark or in the absence of light. It is known dark reaction because it is independent of light. Whether or not light is present, dark reaction can still take place. This means that light reaction will be, light reaction and dark reaction can take place simultaneously. For example, a plant at 12 in the afternoon or 2 in the afternoon where there is abundant sunlight can perform light reaction as well as dark reaction at the same time, because dark reaction does not require darkness for it to occur. It is just independent of light. It can occur in darkness, it can also occur in conditions where light is present. Alright, so this dark reaction occurs in the stroma of chloroplast. Now if you remember from lower classes, this stroma or this matrix of chloroplast is the same thing which is known as actually known as the matrix of chloroplast, the liquid material present in the chloroplast. The cytoplasmic, actually it is not the cytoplasm, the liquid matrix of the cytoplasm, where all the of the chloroplast, sorry, where all the enzymes for photosynthesis are present, and glucose molecules, which will be formed after the dark reaction is complete, is also present in stroma of chloroplast. So dark reaction, the site for dark reaction is stroma of chloroplast. Likewise, the site for light reaction or light dependent reaction is grana of chloroplast. Now dark reaction can take place in a variety of ways. One of the ways which we will discuss in this lecture is the Calvin cycle or the C3 cycle. Alright, there are other ways also, like it can occur through another pathway known as C4 cycle. It is also known as Hatch and Slack pathway for photosynthesis. So actually C4 plants, the plants which perform C4 cycle, in fact better than the plants which solely depend on Calvin cycle for photosynthesis or the dark reaction of photosynthesis. So we'll leave it here because it will take a lot of time for me to discuss C3 cycle as well as C4 cycle in tandem. So first of all, we'll discuss about light dependent reaction in the next step. So light dependent reaction, as you know, are of two types. The first is non-cyclic photophosphorylation, as you can see in the graphic shown to you, and the other variation of the same light reaction is the cyclic photophosphorylation, which we'll discuss in the next slide. Now if you see closely, this complex of molecules is known as the light harvesting complex. You can see this is a molecule termed as P680. P680 forms the photocenter of this photosystem, which is known as photosystem 2. Now photosystem 2 contains the main chlorophyll molecule of the plant, whether it is chlorophyll A or chlorophyll B, and the green discs which are shown all around this P680 or the main chlorophyll molecule are the other molecules of chlorophyll, as well as the accessory pigments like carotenoids, xanthophylls, and the other accessory pigments. Right, so together they constitute the photosystem 2, having the photocenter P680. And similarly, here you see there is another photosystem known as photosystem 1, having the photocenter P700, and around this main chlorophyll molecule are the accessory pigments and other chlorophyll molecules. All right, and on the left hand side you can see the energy of electron. It is lower on this side, lower on the lower side, and higher on the higher side. All right, so this is a photon which is coming from the light source. The light source can be sun or any other artificial source of light. This photon contains light, and as it falls on this chlorophyll molecule or the molecule of accessory pigment, this energy of the photon is captured by the molecule of chlorophyll. Now you can see that every molecule of the photosystem 2 transfers all its energy that it gets from photons to the main chlorophyll molecule known as the photocenter of the photosystem, where it is written P680 in this slide. Right, because of all this energy, electrons present in the chlorophyll molecules get excited. Now you can see 4 electrons here are getting excited and enter the excited state, uh, having higher energy. These electrons at the higher energy are not so stable, so in order to stabilize them, a molecule known as primary electron acceptor or pheophytin, the molecule's name is pheophytin, it is one of the primary electron acceptors of non-cyclic photophosphorylation. Pheophytin grabs these electrons, it stabilizes them only temporarily. Now as soon as pheophytin captures all these 4 electrons, sorry, which were excited after the photon fell on the photosystem 2, this pheophytin gradually releases those electrons. Now these electrons transfer to this molecule known as PQ. PQ is actually plastoquinone, right? So in this transfer, the electrons lose some amount of their energy. As you can see here, the electrons were at a higher energy state, and at the level of plastoquinone, they lose some amount of energy and are present at a slightly lower energy state. Now, plastoquinone does not want all these high energy electrons with it, so it transfers these electrons into the cytochrome complex. Cytochrome complex is a complex of various molecules having cytochrome properties. So, uh, the electrons continue to travel downwards in the energy spiral from the cytochrome complex to another molecule written as PC. PC stands for plastocyanin. It is blue colored pigment or blue colored molecule. So plastocyanin, at the level of plastocyanin, the energy of electrons is lower than as compared to cytochrome complex or plastoquinone. Now here, this is the point where an important thing occurs. This is the point where photophosphorylation is occurring during the non-cyclic part of light reaction. Actually what happens is, when the electrons are moving from plastoquinone to cytochrome complex and from cytochrome complex to plastocyanin, the energy they lose is used by the ADP molecule, that is adenosine diphosphate molecules, in order to phosphorylate. Now adenosine diphosphate or ADP combines with an inorganic phosphate and forms a molecule of ATP, which is known as the energy currency of the cell. This process is known as photophosphorylation. Right, so now you must have understood a little about photophosphorylation. What is photophosphorylation? So that is why it is written ATP is produced via proton motive force. The protons, while moving downwards, the electrons, while moving downwards, release some energy which is used by the ADP molecules in order to phosphorylate themselves and form ATP molecules. Now back to the electrons which are present at the plastocyanin level. The electrons present on in the plastocyanin molecule are lost by it to the photosystem 1, having the photocenter P700. Now, like the photosystem 2, photosystem 1 also acquires energy in the form of photons from the light source. And like in photosystem 2, it also loses some electrons to the excited state. Here, four electrons are lost to the excited state, and again a primary electron acceptor is present here. And then this purple or violet colored molecule is actually ferredoxin molecule. Now this ferredoxin loses these electrons to NADPH. Now it is interesting because you can see here two NADP positive. NADP is nicotinamide adenine diphosphate molecules. These are positively charged NADP positive molecules. And you can see reduction taking place because protons are present here. Now what about electrons? Where do electrons come from? Electrons come from here, where there is photolysis of water taking place. Now two molecules of water break down in the presence of light. The process known as photolysis. Right? These electrons are transferred to the P680 molecules, and thus the electrons which it lost during the excitation of electrons are replenished by water molecules. And in process, four protons are released, and this oxygen is released. This is liberated in the form of gas. So this answers one of our questions, that why is water called the source of electrons and source of oxygen in photosynthesis, or the light reaction of photosynthesis? Because water breaks down to form protons and oxygen. These protons are used by NADP positive molecules in order to reduce themselves to NADPH molecules. Two molecules of NADPH are formed. These will be used during the reduction process of the Calvin cycle or the dark reaction. Right, so non-cyclic photophosphorylation is important because both the photosystem 1 as well as photosystem 2 are being involved. A molecule of ATP is generated, two molecules of NADPH are generated, and photolysis of water takes place. Right, now we'll see in the next slide, photolysis of water is important characteristic of non-cyclic photophosphorylation because it is not occurring in the cyclic part. Now this is the cyclic photophosphorylation. Right, this is the photosystem 1. So the first difference you can see directly looking at the slide is that photosystem 1 is the only photosystem involved in cyclic photophosphorylation. There is no photosystem 2 involved. Right, the process is basically the same. The differences I'll point out later. The photons come to the chlorophyll molecules, and all this energy is transferred to the main photocenter known as P700. It results in the liberation of electrons to the excited state. Here the energy of electrons is higher, so electrons present here are at the excited state. Here a primary electron acceptor molecule accepts these electrons and transfers these electrons to a slightly lower energy state where a molecule known as ferredoxin is present. Right, so electrons now travel to ferredoxin. Now ferredoxin also loses its electrons to a slightly lower state of energy. This molecule we have also seen in uh, non-cyclic photophosphorylation. This is known as plastoquinone. And plastoquinone then transfers its electrons to cytochrome complex. Cytochrome complex uh, also transfers its electrons to plastocyanin, so still lower energy state, and plastocyanin uh, returns the electrons to the photocenter. So here you can see that during the transference of electrons from the plastoquinone through the cytochrome complex and finally to the plastocyanin, uh, an ATP molecule is produced. Right, this is photophosphorylation. Now actually in cyclic photophosphorylation there are 2 points where photophosphorylation is taking place. In this graph it is only shown as 1 point. Let me correct it and check whether it works or not. Right, this is the point where another molecule of ATP is generated. Right, and the transfer of electrons from ferredoxin to plastoquinone, another molecule of ATP is generated. So there are two steps where photophosphorylation is taking place. One during the transference of electrons from ferredoxin to plastoquinone, and the other from the plasticlone to plastocyanin through the cytochrome complex. So there are two points where photophosphorylation is taking place in cyclic photophosphorylation. Now, as you can see, there is no photolysis of water taking place because it does not require photolysis of water, as the electrons the photocenter releases are transferred back by the molecules of this pathway, like plastocyanin returns back the electrons to the photocenter. In the previous slide where we learnt about non-cyclic photophosphorylation, you can see plastocyanin does not return the electrons back to this P680 molecule. Rather, it transfers its electrons to P700. So P680 is devoid of electrons, and those electrons are replenished by the water molecule, the breaking of water molecules. So this was the light reaction part. So the important things in light reaction is that we produce ATP molecules and we produce NADPH in non-cyclic part. Now these are the basic differences between cyclic and non-cyclic photophosphorylation. As you can see, cyclic photophosphorylation, only photosystem 1 is involved, PS1, and in non-cyclic both photosystem 1 and photosystem 2 are involved. Then electrons travel back to the chlorophyll molecule in cyclic photophosphorylation, like we've seen that plastocyanin molecule returns the electrons back to the P700 chlorophyll. While in non-cyclic part, water acts as the electron donor. The electrons replenished to the P680 molecule come from water. No photolysis of water takes place in cyclic photophosphorylation, whereas photolysis of water occurs during non-cyclic photophosphorylation. Then no oxygen is evolved because there is no photolysis of water, and because in non-cyclic photophosphorylation photolysis occurs, thus oxygen is also liberated out. Then, as I've discussed, photophosphorylation occurs at two places during cyclic photophosphorylation, while it occurs only at a single place during non-cyclic photophosphorylation. And NADP is not reduced to NADPH2 in cyclic photophosphorylation, while here in non-cyclic photophosphorylation, this is important, NADP positive is reduced to NADPH2 molecules. This is also known as the reducing part. Right, so these are the basic differences between cyclic and non-cyclic parts of photosynthesis. Now, during this light reaction, we have produced ATP and NADPH molecules, which will be used in the next phase of the reaction, known as Calvin cycle or the dark reaction. Here is the process of Calvin cycle. As we've learned earlier, the Calvin cycle occurs in the stroma of chloroplast. Right, so what occurs in the stroma? This the site where this reaction is taking place. It's written all the three phases of the Calvin cycle take place in the stroma of chloroplast. Now, what are these three phases? The first phase is fixation. Right, fixation. It's written in the reaction: 3 RuBP molecules plus 3 carbon dioxide molecules form 6 molecules of 3-phosphoglycerate. I'll come to that in this part B, where the reaction is described. The second part is reduction. Now this phosphoglycerate molecule which is formed will be reduced, and in order to reduce this, we'll require the reducing part formed during non-cyclic photophosphorylation, known as NADPH, and some ATP molecules will also be used. Now 3-phosphoglycerate molecules are reduced in the presence of ATP and NADPH molecules to 5 molecules known as G3P, G3PR glyceraldehyde 3-phosphate, and 1 molecule of G3P goes further to yield glucose or fructose. And finally, G3P or glyceraldehyde 3-phosphate, 5 molecules of this use 3 molecules of ATP in order to produce 3 molecules of RuBP. Now this might seem somewhat confusing. I'll try to make it more clear. Now focus on this reaction here in this cycle. This is the first step known as fixation of carbon dioxide. You can see a molecule here, RuBP. All these 5 dots represent carbon molecules. It is a biphosphate. It is actually a ribulose biphosphate molecule. 2 phosphate groups are present at either ends of this carbon chain. Ribulose is a 5-carbon sugar and it is phosphorylated, so it is ribulose biphosphate. Right, this is RuBP molecule, and here we can see 3 carbon dioxide molecules coming to join with 3 molecules of RuBP. Now, 3 molecules of RuBP will contain 3 into 5, total of 15 carbon atoms, and each molecule of carbon dioxide contributes 1 carbon atom to the reaction. So 15 from here and 3 from here, together we get 18 molecules of carbon, right? So after this first reaction, 6 molecules of this 3-phosphoglycerate are formed. Now 3-phosphoglycerate molecules are molecules having 3 carbon atoms each. So 6 into 3, 18. We had initially 18 carbon atoms, so the number of carbon atoms is still the same. 18 carbon atoms are with us in the form of 6 molecules of 3-phosphoglycerate ions. This Calvin cycle is also known as C3 cycle because this 3-phosphoglycerate is the first stable product of this cycle, and you must remember this. This is one of the important questions. What is the first stable product of Calvin cycle? The answer is 3-phosphoglycerate. So now this is the first step of this cycle called fixation of carbon dioxide. It is also known as carboxylation of RuBP, and it requires an enzyme known as rubisco. Now rubisco is important because it is the most abundant protein on earth, so you also should remember that. Now we'll move on to the second step of this reaction. Second step is reduction of this 3-phosphoglycerate to form glyceraldehyde 3-phosphate, right? In order to reduce these molecules, for each molecule of 3-phosphoglycerate will require 1 molecule of ATP and 1 molecule of NADPH. We had 6 molecules here, so 6 molecules of ATP are converted to 6 molecules of ADP, and inorganic phosphate is liberated, and 6 molecules of NADPH are oxidized to form 6 molecules of NADP positive, and 6 protons are released. Now, as it is known as the reducing agent, it reduces 3-phosphoglycerate molecules to molecules of glyceraldehyde 3-phosphate. Now these molecules also contain 3 carbon atoms, right? So 6 into 3, we still have 18 carbon atoms with us. Now you can see a downward reaction going here. One molecule out of these 6 molecules of G3P or glyceraldehyde 3-phosphate goes in a downward reaction where it forms glucose, right? Actually 2 molecules of these in 2 different cycles would be required to form 1 molecule of glucose. Now we are left with 5 molecules of G3P here. Now this brings us to the third step of Calvin cycle, known as regeneration of RuBP from G3P, that is glyceraldehyde 3-phosphate. What happens here is, in order to regenerate 1 molecule of RuBP, you require 1 molecule of ATP. We started with 3 molecules of RuBP. We have to regenerate 3 molecules of RuBP, so we require 3 molecules of ATP. So you can see 3 molecules of ATP are getting used up to form 3 molecules of ADP, and 3 inorganic phosphates are released. You can calculate from here. 5 G3P molecules will have 5 into 3, 15 carbon atoms, and 3 molecules of RuBP will have 3 into 5, 15 carbon atoms. So it is basically a rearrangement of carbon molecules, as you can say in layman's language. Like regeneration occurs. Sorry, got a little blurred. Right. Regeneration occurs from G3P, that is glyceraldehyde 3-phosphate. Now the summary of this entire reaction is that 6 here, 3 here, right? So for every 3 molecules of carbon dioxide, 6 and 3, 9 molecules of ATP are utilized, and 6 molecules of NADPH are utilized. How many carbon atoms does a glucose molecule have? The answer is 6. So in order, because RuBP is regenerated entirely, the only thing which is not regenerated is carbon dioxide. That means the carbon in glucose molecule, here the glucose molecule is getting formed, the carbon in glucose molecule is coming from the molecule of carbon dioxide. So each time a molecule of carbon dioxide enters this cycle, 1 molecule of carbon or 1 atom of carbon enters the glucose. So in order to form 1 complete molecule of glucose, you have to turn this cycle 6 times, or 6 times the carbon dioxide has to enter the reaction so that a molecule of glucose or fructose can be formed, right? So it leads to another question: how many ATP molecules are required to form a molecule of glucose? The answer would be 18, because For 3 carbon dioxide molecules, we required 6 plus 3, 9 ATP molecules. For 6, we'll require 6 plus here would be 12, 18 molecules of carbon dioxide, ATP. And similarly, 12 molecules of NADPH would be required in order to produce 1 molecule of glucose. So this is the summary of the photosynthetic process involving the light reaction and the dark reaction. I hope you understood it properly, and I hope my communication was good enough for you. So in the end, I just want to say thank you for listening, and have a good day.